Hey there, welcome. Welcome to the Middle Author Bioinformatics Microbial Omics app. This video is just to give you a little bit of a sort of lay of the land around here and how things work. So we're going to start with this landing page, and this is where you'll end up if you go to this address, omics.midauthorbio.com. So on this landing page, you'll see a series of grids, and these basically just represent the software tools that are available on this web app. And these are all open source currently. And if you want to check these tools out, just go ahead and click on any one of these tiles and it'll take you to the Wikipedia or actually the GitHub page for each individual tool. And there you'll find more information about how it was designed, programmed, and published. So this is an academic version of the web app. There are certain limits you have to abide by. For example, if you want to upload FASTQ files for analysis, the limit there is 1 GB per FASTQ file. Now that includes gzipped files, which you can also provide. So that gives you a little bit more room. Metagenome assemblies, also 1 GB per metagenome assembly file. And if you're providing prokaryotic genomes for analysis, the max there is 10 megabytes per genome file. So if you have more files, more data you want to use, please don't hesitate to reach out and we can set something up custom for you. Tab navigation is pretty straightforward. You can scroll to the right, to the left. As we're adding more tools, you'll be able to scroll further to the right. If you click on any of these particular tabs, it'll take you to the submission form where you'll find that you're only required to provide two pieces of information. That's your name and your email, where the results of the analysis will be emailed to. And of course, you're going to have to upload your file here. The specific file types for each individual software tools are sort of described here. For example, Spray and Pray will accept only FASTA files. So you got to know what those are. And then you got to select whether you have a genome or a metagenome. Same for other tools. MegaHit accepts FASTQ files. Pseudofinder accepts GenBank files. So those are files that end in .gbk or .gbff or .gb. Currently, GitaTree has the more complicated set of files that it accepts, and it accepts all types of files, protein files, GenBank files, and raw genome assemblies, or also a single column file of NCBI assembly sessions. And those are the ones that start with GCA or GCF. So once you submit your data and you put your name and you put your email in, once the data is received by our server, you'll get an email saying that the data is received and the analysis has begun. And then all you got to do is just wait around and wait for your results to get there. Now, of course, once the results do get there, the data is wiped clean. We don't keep any data, so you don't have to worry about your data being used for anything. And we do want to emphasize that this is for research and educational use only. So it's not intended for any sort of clinical or diagnostic purposes. So if you are interested in that, please do hit this contact us button and reach out. A few last things. I have a music icon here. If you want to set yourself up with a little bit of ambiance, you can click this and hit the play button, and that'll create a little YouTube window here with a nice fire crackling and some classical music playing. If you want to subscribe to an email list, since we do offer pretty regular updates, and we're always trying to improve things. So put your name and email if you have a comment or suggestion, put that in here, and then just hit the subscribe button, and you'll be subscribed. Thanks again for checking out this app, and good luck.